It survived the vacuum of outer space. It endured crushing pressures that would destroy a submarine. It shrugged off radiation 1,000 times beyond what would kill any human. And it did all of this with a body built from roughly 1,000 cells. The tardigrade is the most indestructible creature on Earth, and possibly the most extraordinary organism that has ever existed. But what scientists recently discovered inside its DNA is not just rewriting biology. It could one day make humans nearly indestructible too. To understand how, you have to go back 500 million years. 500 million years ago, before the first fish swam or the earliest ferns unfurled, a creature smaller than a grain of sand was already exploring the raw edges of survival. The tardigrade belongs to its own phylum, Tardigrada, a distinction reserved for evolutionary lineages as ancient as insects or crustaceans. Fossils pressed into Cambrian shale, dated to around 540 million years ago, reveal impressions that match the body plan seen today. A compact, segmented form, armored in a flexible cuticle with 4 pairs of stubby, claw-tipped legs. Each adult tardigrade is divided into 5 segments, a simple blueprint that has endured since the dawn of complex life. Size alone defies expectation. Most tardigrades are less than a millimeter long, about the width of a human hair. Hatchlings can measure just 50 microns, vanishingly small yet already complete. Despite this minimalism, every tardigrade packs the machinery of survival into roughly 1,000 cells. By comparison, a fruit fly embryo contains tens of thousands. Yet, in the world of the tardigrade, less is more. The streamlined anatomy means fewer points of failure and an extraordinary ability to control every vital process, from movement to metabolic shutdown. Under high-powered microscopes, the tardigrade's details emerge in stunning clarity. The claws, sometimes shaped like tiny grappling hooks, anchor the animal to moss, lichen, or the mineral crust of a hydrothermal vent. The cuticle, layered and sometimes decorated with spines or plates, shields the soft tissues from desiccation and crushing pressure. The mouth, ringed by a muscular pharynx, pierces plant cells or devours smaller invertebrates. Every structure is a product of relentless adaptation, honed by eons of environmental upheaval, volcanic eruptions, asteroid impacts, the rise and fall of oceans. This is not a fragile organism clinging to the margins. The tardigrade is a veteran of planetary catastrophe, a survivor whose lineage predates dinosaurs, trees, and even the earliest vertebrates. Its design is not just a curiosity of nature, but a toolkit assembled for resilience. By tracing its physical form and evolutionary journey, the outline of an indestructible survivor comes into focus, one that has outlasted nearly every other branch of complex life on Earth. A tardigrade's greatest weapon is not its claws nor its armor, but its ability to vanish from life's demands altogether. When water disappears or the environment turns hostile, the animal initiates a transformation so radical it borders on science fiction. The process begins with a slow withdrawal. Legs retract, the body contracts, and internal fluids are expelled. What remains is a compact capsule called a ton, where every cell is locked down, every system placed on hold. Metabolism plummets to less than 1/10,000th of normal. In this state, the difference between life and death is almost academic. For all practical purposes, the tardigrade is paused, but this is not mere dormancy. Inside the ton, a chemical metamorphosis unfolds. Water, the solvent of life, is replaced by a molecular glass. The disaccharide tree, halose, floods the cells, weaving itself among proteins and membranes. As dehydration deepens, tree halose molecules interlock, forming an amorphous matrix that stabilizes everything it touches. Alongside tree halose, a unique family of proteins called CAHS spring into action. These proteins are shapeshifters, disordered and flexible in solution, but as the cell dries, they self-assemble into a dense, gel-like scaffold. The CAHS trihalose synergy is essential. Trihalose alone can slow down molecular chaos, but with CAHS, the cytoplasm vitrifies, a true glass transition where movement ceases and biological time nearly stops. This vitrified matrix is more than a shield against dryness. It immobilizes enzymes, preserves the delicate architecture of DNA, and prevents the fusion of cell membranes. No ice crystals form, no proteins unravel. Even the mitochondria, the cell's powerhouses, are held in perfect suspension. The ton can endure years, even decades, in this state, awaiting the return of water. Then, with a single drop, the glass dissolves, CAHS proteins relax, and the tardigrade resumes life as if nothing happened. The secret of the ton is not brute strength, but a biochemical switch, a reversible shutdown that rewrites the rules of survival. By vitrifying themselves, tardigrades evade the damage that kills nearly all other forms of life. This ability is not just a curiosity, it is the foundation for every extreme feat that follows, from surviving space vacuum to enduring lethal radiation. The power to turn off, to become almost nothing, is what makes the tardigrade the most indestructible creature on Earth. In September 2007, a Russian capsule streaked through the upper atmosphere, returning from a journey that rewrote the boundaries of survival. Inside, sealed behind a thin glass window, were several hundred tardigrades, each one locked in a cryptobiotic ton, dormant and dry, awaiting the impossible. For 10 days, these animals had orbited Earth aboard the Photon M3 satellite, exposed to the raw vacuum of space, solar ultraviolet radiation, and wild temperature swings from -20 to 40 degrees Celsius. There was no atmosphere, no water, no shield but their own molecular defenses. The experiment, led by Dr. Ingemar Jonsson, was simple in its audacity. Could a multicellular animal survive direct exposure to outer space? When the capsule was opened and the tardigrades rehydrated, the answer stunned even the researchers. More than 99% of those exposed only to vacuum revived, stretching out their legs and crawling as if nothing had happened. Some had also faced filtered ultraviolet light, and a smaller fraction, about 68%, returned to life. Only a handful survived the full blast of unfiltered solar ultraviolet, but even these few proved that life could persist where none was thought possible. Dr. Johnson described the moment. Seeing those tiny bears wake up after a trek through vacuum was like watching a phoenix rise from nothingness. The survivors not only moved, they reproduced, laying eggs that hatched into healthy young. No visible DNA damage, no loss of structure. The tardigrade's ability to pause all metabolism, vitrify its cells, and shield its genetic code had worked even in the emptiness of space. This was not a fluke or a trick of the laboratory. It was a demonstration that cryptobiosis is a universal defense, effective even beyond the planet that shaped it. For the first time, scientists had proof. The most indestructible creature on Earth could also survive the void between worlds. Boiling water hisses over volcanic rock, and in the shadows of geothermal vents, life is put to the test. Most organisms unravel in the face of such heat, but the tardigrade endures. Laboratory trials have pushed these animals to 150 degrees Celsius, hotter than a kitchen oven. For up to 30 seconds, dehydrated tons withstand this searing pulse, their internal machinery locked in a glass-like matrix that refuses to melt. In nature, tardigrades have been found clinging to mosses on scalding geothermal crusts, surviving brief brushes with temperatures that would cook almost anything else alive. Pressure, too, poses a threat. Deep beneath the ocean's surface, where sunlight fails and the weight of water builds relentlessly, few animals can persist. Yet, in 2020, researchers at Riken in Japan subjected Rhamazotius varieornatus to a crushing 1.2 gigapascals, 12,000 times atmospheric pressure, equivalent to the weight at the bottom of a 12-kilometer deep trench. 85% of the specimens revived after a full day under these conditions, stretching out their legs as if nothing had happened. The tardigrade's flexible cuticle, paired with internal proteins that act as molecular chaperones, prevents the collapse of membranes and the denaturation of vital enzymes. Among these defenders, the HSP70 heat shock protein surges in abundance, helping to refold proteins and maintain cellular order when the environment turns hostile. Heat and pressure are forces that warp and destroy, yet for the tardigrade, they become just another set of boundaries to ignore. The secret lies not only in physical toughness, but in a suite of molecular adaptations, proteins that stabilize, chaperones that repair, and a body plan that bends but does not break. Each extreme conquered adds to the legend of the indestructible water bear, expanding the list of environments where life, against all odds, refuses to yield. Nuclear radiation tears through living tissue with a violence that leaves few survivors. For humans, a dose of just 5 gray is usually fatal. Cells rupture, DNA shatters, and organs fail within days. Yet, in laboratory tests, tardigrades have endured gamma radiation at levels 1,000 times higher, up to 5,000 gray. At this intensity, every strand of DNA is broken many times over, proteins unravel, and even the strongest bacteria perish. But in the cryptobiotic tun, the tardigrade waits out the storm. Most animals would be erased by such punishment. Here, a handful revive, sometimes after days of apparent lifelessness, crawling forward as if nothing had happened. The scale of this feat is almost impossible to grasp. The Hiroshima bomb released radiation doses of about 4 gray at ground zero. A tardigrade could withstand the equivalent of 1,000 bombs of gamma exposure and still return to life. No vertebrate, no insect, not even the hardiest spore-forming bacterium comes close. The survivors may move slowly at first, but their vital machinery, including the nucleus, mitochondria, and cell membranes, remains intact. Eggs laid before irradiation may still hatch. DNA damage, visible as molecular scars, is rapidly repaired in the hours after revival. This is not just resistance, it is a redefinition of what life can endure. The tardigrade's molecular defenses, honed over 500 million years, allow it to shrug off a threat that defines the boundary between life and death for almost every other organism. In the world of radiation, the tardigrade stands alone. The question that follows is not just how it survives, but what secrets lie within its DNA. Secrets that may hold the key to protecting life itself from the most lethal forces in the universe. In 2016, Professor Takakazu Kunieda's team at the University of Tokyo isolated a single gene from the tardigrade, DSUP, or the damage suppressor, and changed the way scientists thought about DNA protection. When DSUP was inserted into human embryonic kidney cells, the results were startling. Exposed to 10 gray of ionizing radiation, 95% of these modified cells survived, while unaltered cells died off. DSUP forms a loose, filamentous shield around chromatin, blocking the reactive oxygen species that would otherwise tear through DNA. Follow-up studies confirmed this was not an anomaly. The gene's effect was robust, slashing double-strand breaks by nearly half and offering a glimpse of what true radiation resistance might look like in human biology. The breakthrough quickly moved from cell cultures to broader experiments. CRISPR and lipid nanoparticle delivery methods enabled DSUP expression in various mammalian cells. In mice, a single dose of DSUP-MERA led to much higher survival rates after whole-body irradiation. When human stem cells were engineered to express both DSUP and a tardigrade version of RAD51, a DNA repair enzyme, the cells endured up to 15 gray, a dose that would destroy any ordinary cell line. The synergy between DSUP's shielding and RAD51's repair mechanisms created a dual defense, setting a new standard for biotechnological resilience. Yet the promise of DSUP is shadowed by uncertainty. Professor Cañeda, while optimistic, warns that translating these results to living humans is fraught with risk. Off-target genetic effects, immune responses, and unpredictable consequences have led bioethicists to urge caution. Regulatory agencies in the US and Europe now classify DSUP-modified human cells as gene therapy products, requiring strict safety trials. The debate has shifted from theory to urgent reality. The idea of engineering humans to survive nuclear fallout or deep space is no longer distant speculation. As biology and technology merge, the tardigrade's resilience may soon belong to us, forcing a reckoning with the true cost of survival. A single tardigrade, sealed in a glass capsule, can endure the kind of shock that pulverizes rock and launches meteorites across space. In laboratory simulations of panspermia, the theory that life can travel between planets on fragments of stone, dehydrated tons have been blasted with impact shocks of 10 gigapascals, the same pressure unleashed by a kilometer-wide asteroid. After the violence, researchers rehydrated the survivors. Over 70% revived, stretching their legs as if nothing had happened. This number is not a fluke. It is a data point that narrows the gulf between speculation and possibility, suggesting that microscopic life, locked in cryptobiosis, could survive the chaos of planetary ejection and interplanetary flight. The search for life beyond Earth now turns to the tools that can reveal such survivors. On Europa and Enceladus, icy moons with hidden oceans, the next generation of probes will sample plumes and surface ice, hunting for signatures that echo tardigrade biology. Dr. Lisa Kaltenegger, an astrobiologist, describes the stakes. If a water bear can endure a vacuum, we must keep an open mind about life thriving beneath Europa's ice. Instruments like cryo-Raman spectrometers are being designed to look for the telltale signs of tree halose glass, a sharp spectral peak at 1,730 inverse centimeters. This marker, invisible to the naked eye, could reveal the presence of cryptobiotic life in a frozen world. Microfluidic chips, inspired by the tardigrade's own revival process, may one day rehydrate particles collected from alien oceans, coaxing dormant organisms into motion for the first time in millions of years. The story of the tardigrade is no longer confined to Earth's extremes. Its biology has become a blueprint for imagining what life might endure and how we might find it across the solar system. Each experiment pushes the horizon of possibility a little farther, leaving open the question: if a tardigrade can survive the journey between worlds, what else might be waiting, paused in the darkness, for the right moment to begin again? Every day, scientists push deeper into the tardigrade's molecular mysteries, chasing secrets that might one day shield astronauts, heal human cells, or redefine the limits of life itself. In a universe hostile to survival, this microscopic titan stands as living proof. Resilience is not only possible, it is already real. The most extraordinary creature on Earth may yet shape the fate of worlds beyond. Wonder endures. Thanks for watching.